It's really a great honor to receive this prize, the Kabiller Prize in nanoscience and nanomedicine. I work more on the nanomedicine side. We've heard from some other aspects of nanoscience. Very, very interesting. So I'm really deeply honored and pleased to share a few thoughts with you this afternoon. A couple of comments before I jump into the slides. In the video it was mentioned biologic approaches and chemistry, but actually probably 2/3 of my lab is focused on those biologic approaches. So we have done and do a fair bit of engineering of the matrix. I was really stimulated to hear Viola Vogel's talk today. Super long-standing, profound impact in understanding the matrix in biology, and we've tried to build on that in the context of regenerative medicine, in the context of immuno-oncology, with protein engineering approaches, as well as in the context of biomaterials. So, but today, for this talk, I'll focus on a couple of more chemical examples because I think they're more chemistry-oriented people here in the audience. That's one comment. Another comment is I feel very uncomfortable. So I was asked to give a kind of a high-level talk that could be at least part of it understood by a lay audience, and I feel much more comfortable hiding down in the deep data. So I'm going to try to come up for air a little bit in the first part of the talk with some generally accessible materials, and then I will show a couple of examples with actual data. So with that, what do I mean? Nanotechnology guiding immunology. Guiding what? So like what do I mean by guiding? So guide from what to what? You go from A to B. Is something bad? No, something's bad. I'll continue on. You guide from something. You don't just guide, but you guide from a position to a position. So what I'd like to do is to think about some, in this overview, high-level part, what are some of the challenges in nanotechnology and in immunoengineering in general where we want to guide an immune response. So there, not yet. Okay, there we go. So where we want to guide. So guide from what to what. So in the context of infectious disease, clearly, you want to, partially, you want to guide from a naive state to protect yourself from an organism that you've never seen before, to be a prepared state. Not an actively responding state, but one that has memory that can then respond to a new immunological challenge. That's in the context of prevention of infectious disease. That's, of course, a vaccine and maybe some other things too. And we've had the pleasure of being able to work in the context of nanotechnology in vaccines. I won't show any of those sorts of results. A much more challenge, a larger challenge actually, is in therapy in infectious disease. So meaning from a state of immune suppression to like a chronic viral infection, to a state of an active immune response without making you blow up to all of your infected cells that you like to be able to kill to ameliorate that infection. But so going from a suppressed state to an aggressive state. I'll have a slide on each one of these bullet points here. So in cancer, many of us have had tumorous cells in us, but we've killed them and we didn't know it. Because they were recognized immunologically. But the successful tumor is an immune suppressive tumor. So meaning it overwhelms the immune system. So we want to guide from a suppressive state onto an aggressive state, an autoaggressive state. We work a fair bit, and I'll show an example there. So in the context of allergy, for example, we're exposed to all sorts of antigens that are harmless in the food that we eat, in the air that we breathe, in the bugs that are on our pillows, all sorts of places. So how do we go from a state of triggered, accidental maybe, aggression back to a state of maybe regulation, or at least ignorance? And I'll show an example of that in there. So our lab is quite interested in autoimmunity. And so there is an autoaggression, a self-aggressive response. And we like to go not just from self-aggression to ignorance, but self-aggression to regulation. So this field of tolerance is not just to not attack something, it's rather to protect something. And I'll come to that in some more cases as well. So also there's a fair bit of work in nanotechnology guiding what goes on in the microbiome. So the microbiome, not only in the gut, but maybe most of the targets are in the gut, to maintain homeostasis in cases of dysbiosis that would lead to food allergy, to inflammatory diseases of the bowel. And the microbiome is kind of everywhere now, so affecting everywhere. So a clear gut-brain axis, for example, and not only that. So how do we go from a dysbiotic state to a cooperative state between you and the organisms that you host? So in this high-level bit, I'm going to give like 4 slides of overview of kind of some of the immunological bases of an innate immune response that's very rapid response, and a rapid response that can be protective or can indeed be somehow regulative. In the middle, then, there's an innate component of antigen-presenting cells that collects antigens and presents them to the adaptive immune system. In a vaccine like we got for COVID, you want it to present to induce an expansion, to contract to memory, to have memory B cells and long-lived plasma cells in the marrow, being able to regenerate those antibodies, and importantly, to generate a cellular immune response, not just antibodies, but a cellular immune response. But in the case of tolerance, you want those same antigen-presenting cells, the very same ones, maybe a few others, and I'll mention that, you want them to be able to collect antigen and to turn on regulatory responses. So they're traffic cops deciding which way things go, up or down. And so immunity is really adaptive immunity. The right-hand side of that slide is really context dependent. And so here, if you think of like in the blue, a T cell, a T lymphocyte, and interacting with an antigen-presenting cell, there's a primary signal of a T cell receptor interacting with a peptide loaded on a major histocompatibility complex molecule. There are co-stimulatory molecules that then lead to activation after that engagement. But there are many, many regulatory molecules around which other Nobel Prizes have been won. Other regulatory molecules that maintain immunity from exploding. So when you encounter a virus, so you feel things like your lymph nodes get larger, swell. That's because there's a lot of T cell proliferation and B cell proliferation going on in the lymph nodes. But you sort of without a restraint, you would turn into one big bag of pus. So that is contracted, and that's contracted very actively by some of these regulatory molecules like CTLA4 and PD1 and PDL1. So that leads to inhibitory responses, which are beneficial in the case of tolerance, beneficial in the case of reacting to inflammation, and other ways as well. So in the context of the left-hand side of activation, that's all good news in the case of pathogens and tumor antigens, which are mutated proteins. And on the right-hand side, on the inhibition side, that's all good news in the case of tissue repair. There are adaptive immune responses involved in tissue repair, or responding to commensal organisms, or to food antigens that you would have encountered. But when that runs amok, then you can get activation spreading over to where inhibition would have been, leading to autoimmunity and allergy. So these checkpoint molecules, CTLA4, PD1, PDL1, they're the whole alphabet soup of them now. They're all prominent targets in the context of cancer. So in cancer, those checkpoint molecules are harmful. In regulation, they're helpful. They were there to be helpful, and the smart cancer figured out that, upregulated those molecules, and uses it to escape immunity. So people try to block that, and that was a Nobel Prize a couple of years ago. So in the context of autoimmunity, then they're helpful to indeed resorb. This was one of the slides in the little video, but yeah, tipping the balance. So I really am lucky to have a lab that thinks about both immuno-oncology and inflammation, immunity, allergy, autoimmunity, because to us, it's all the same thing. It's maybe the details are a little bit different, but it's the same thing of a balance between immune aggression, sometimes I want it, sometimes I don't want it, and immune tolerance, sometimes I don't want it, sometimes I do want it. But we find that what we understand on one side educates us on the other side. So I'd just like to go, still in this higher level introduction. By the way, I tried to ignore those emails to keep it high level in the beginning until the 8th one, and then I decided I better pay attention. So in the context of infectious disease, what are some of the challenges there that are left? I mean, some vaccines work really well. The malaria vaccine works poorly. The HIV vaccine doesn't exist. So there are details there. But not only among those details, but how do we vaccinate economically and with durability? Many vaccines aren't so durable, and one needs to, like the malaria vaccine, is not very which is not terribly successful. It's approved, it's licensed, but it's not terribly durable. So, in the context of global healthcare, so we all, most of us, got the mRNA vaccine, one or the other. That made very little impact in global healthcare, global care, because of cold chain storage. It's hard enough to have a refrigerator, much less a freezer, much less a -80 freezer. So how do we do that in the context of something that can be stored at room temperature, which for mRNA probably means dry too. So there are 1,000 details there. And I'd just like to mention, although the Nobel Prize was given for the mRNA part in that chemistry, but there was like 25 years of pre-work on the lipid nanoparticle technology, and 1,000 details there that went into that. So one of the aspects of nanomaterials in the context of infectious diseases has to do with their size. And I wanted to mention some really old work just to think about why nano, what's so special about it there? Why couldn't you make it micro or whatever? A particular target is the lymph node. The lymph node is like Grand Central Station, where T cells and B cells and dendritic cells and antigens and other molecules and other cells are present there, tailoring these immune responses. And the question is, how do you get it to the lymph node? This is a diagram on the left-hand side from one of Melody Swartz, my longtime vaccine collaborator and cancer vaccine collaborator, thinking about interstitial flow that exists between a blood capillary and a lymphatic capillary. And that if we can make a material that was sufficiently small to be dragged along in that interstitial flow, which is order of magnitude 0.1 to 1 a micrometer per second, then that could be convected into the draining lymphatics, cleared lickety-split into the lymph node. The image on the right-hand side was collected by Cy Reddy. I mentioned students who dragged me in immunology. Cy was the first one of those. He's now a professor at ETH Zurich. Super strong in immunoengineering. But you'll see an injection site in the red in the foot pad. But you'll notice, I'm not sure if I can point with this. You see the pointer? No. So you'll notice, for those on the video not going to see that, but you'll notice down here, so that's the back of the knee, and that's the popliteal lymph node. That's like 2 centimeters away from the foot pad. That image was taken after 10 minutes. So diffusion had absolutely nothing to do with that transport process. That was all convection. And if you look at the details of that convection, this is from a 2006 paper in the journal controlled release. How nano what? How big, how little, I don't know. But like if we make 100 nanometer particles, that drainage is very, very slow. But at 25 nanometers, 30 nanometers, that drainage is very, very fast. These are images of a lymph node, that one behind the knee, a couple of centimeters away from the foot pad. So you see in the red is the nanoparticles, but high accumulation, but strong size dependence, where the micro size is totally wrong. Even like kind of larger in the nano size isn't very good either, but much larger. Gets us to Grand Central Station for immune recognition. That's just a little vignette of how the nano dimension helps us in vaccines by getting to these target sites like the lymph node. So we went on, and Cy Reddy then published a 2007 paper in Nature Biotechnology showing this in a full-blown vaccine context, and that nanomaterial vaccine life has stayed with the lab in a number of iterations. I'd just like to mention a challenge that is not yet successfully addressed in infectious disease, from suppression to aggression. It's nice to think of what I could do if I could do something. A bit like 300 million people in the United States, in the world, not United States, that's the size, but 300 million people in the world are infected with hepatitis B chronically. Not acutely, but chronically. They fail to clear it. They have it forever. It leads to cirrhosis of the liver. It leads to hepatocellular carcinoma. It leads to about 1 million deaths annually. And the vaccine that many of us have received, maybe most of us, does nothing at all. It's a therapeutic vaccine. It doesn't do it's a prophylactic vaccine. It doesn't do anything from therapy. So there are major challenges for chronic viral disease. HIV is an enormous one for which there's absolutely no success. But human papillomavirus, there are many others that you'd like to be able to address. I'd like to mention cancer. What do I mean from suppression to aggression? What is suppression there? What is the successful tumor doing that is suppressive? By definition, the tumor has mutated proteins. That was mentioned in the previous talk. They're thinking about mutations in KRAS. By definition, those mutated proteins were not tolerized centrally in the thymus. So by definition, they are neoantigens. So you should be able to have an immune response to those. So the tumor is kind of tricky. It can downregulate the expression of some of those neoantigens if you attack them and thus escape. It can downregulate its expression of MHC1 and not present what's inside and thus hide. And it can upregulate those checkpoint molecules like PD-L1, which people drug with antibody drugs. So we'd like to be able to ask, how do we use nanotechnology to block those things? So to block downregulation of MHC1, to mean immune stealthiness. If I just don't show my feelings, meaning the antigens that are inside, if I don't show them on a surface, they can't be attacked. So how do I block that? For example, using sRNA-based knockdown in tumor cells. In allergy, I'm going to show an example on allergy, and I'll come back to this slide. But the question is in the context of pre-existing immunity. I have allergy. How would I protect and undo that allergy? Right now, there are therapies. The therapies are a subcutaneous injection, mostly of allergens from pollen mixtures or house dust mite mixtures, or sometimes food mixtures. It's a very low efficacy, super demanding, very long therapy to desensitize a patient. But we and others are asking, how do I use a nanotechnology approach to more, in a pointed, mechanistically definable way, undo immunity? How do I reverse it? But I'll show you an example of that. We're very interested in autoimmunity. So going from this self-regulation to, self-aggression to self-regulation. So, but I'll come to that again. And the question is, there are many autoimmune therapies. You see them when you watch TV at night, targeting IL-17, targeting IL-23, targeting many molecules that are inhibitors. The huge one targeting TNF-alpha in the context of rheumatoid arthritis, the drug Humira, which is the largest drug that ever was. So those are inhibitors of pro-inflammatory agents. But the thing is, you have those pro-inflammatory agents for a reason. So if I inhibit them, that means I systemically suppress you. And we would rather be able to do that in a way that did not involve systemic suppression. When you hear those ads, there's a guy that talks really fast at the end about all these horrible things that might happen to you. You might die, and that's all because of the immune suppression. There can be even latent viruses that reestablish when you take the brakes off. The last thing I'll mention before I go on to a couple of specific examples is think about the microbiome. And there, nanotechnology plays a role too. So a microbiome, people have tried very hard for the last 10 years with many companies, some come, some gone, a few still there, that have tried to identify the right bugs and to repopulate the gut with the right bugs. That turned out to be quite a heavy lift. Most of those bugs are very sensitive to oxygen, and getting them to reestablish in the distal gut where there's very low oxygen turned out to be a major challenge. So an approach for nanotechnology, and I'm not going to show you this example, we published it in 2022 in Nature Biomedical Engineering with my colleague there, Catherine Nagler. She did the immunology there, we did the chemistry. But asking the bug, okay, what about the bug? What does the bug do? Many things probably, but what it does mainly is produce metabolites. And those metabolites are signaling molecules that we evolved with. We co-evolve with that. And so they're very important immune regulators. One of them is butyric acid. Butyric acid is very low. It's an HDAC inhibitor. There's a GPCR, a couple of them that are signaled by butyric acid. But it's all quite low potency. And so you can go buy from Amazon capsules of butyric acid. They smell awful. I tried them. They taste really bad too. But they don't do anything either. So you would, for the dose of it you would need, you're taking like 30 pills a day. And it has to be in a pill because it tastes so horrific. So what we ask is, can we make nanomaterials? Can we make polymer micelles? A polymer micelle that contains linked to a domain of the polymer, butyrate, that can be cleaved by esterases in the distal gut to replace the function of the dysbiotic microbiome and make it look normal biotic. And so this paper by Ruyi Wang and Shiji Cao, as I said, in close collaboration with Kathy Nagler's lab, we were able to show prevention of food allergy in responses in food allergic mice, and also a favorable response in a colitis model, an inflammatory disease model. But I'd like to go on to two examples and actually mention something about specificity. Something that we started working on, about 2013, are called inverse vaccines. So meaning, we all know what a vaccine is, but a vaccine that could unravel something, that could induce a regulatory response where there's a pre-existing effector and memory response, autoaggression, and I want to undo it. To be clear, we did not invent the concept of an inverse vaccine. We didn't invent the word either. I think the word was invented by Lawrence Steinman at Stanford in about the year 2010. We've just sort of taken a different chemical approach to it that seems to be rather promising. So what is a vaccine? So we see here a first wave of expansion in the blue, a first wave of expansion. And when you're re-exposed to the antigen, so like an infection after the vaccine, then you expand again and you kill it. So in an inverse vaccine, we deliver an antigen, so meaning it's antigen specific, just like a vaccine. That's why we call it a vaccine. It's antigen specific, but we expand a population that leads to the induction of regulatory T cells and maybe the removal of autoaggressive cells by apoptosis, or so-called allergy. Such that when you're re-exposed, like antigens in my brain or antigens in my spinal cord that are there all the time, when I'm re-exposed, I don't respond to them, for example, in multiple sclerosis. So we were particularly interested in antigen processing in the liver. And here I really acknowledge Scott Wilson, a fantastic bioengineer, chemist in the lab who's now running his own lab at Johns Hopkins. And so we together started to think about the liver and the role of liver in maintaining peripheral tolerance. So what do I mean by peripheral tolerance? Central tolerance happens in the thymus. And the thymic tolerance is leaky, it's called leaky, meaning it doesn't perfectly delete or tolerize against self-antigens. So for example, vitiligo, the autoimmune disease that leads to skin depigmentation, everybody in this room has autoaggressive T cells to that protein in skin that could cause vitiligo. But we regulate it. Those autoaggressive T cells, they're there in quite high frequency, but they're regulated, and therefore we don't develop the disease. So that's what I mean by what is peripheral tolerance. It means tolerance not happening That was peripheral. They're not happening in the thymus, but happening extrathymically, in the lymph nodes, in the liver, those are probably, in the spleen, those are probably the sites. Why does it exist? We've all studied a little bit of biochemistry and thought about post-translational modifications. Most of those post-translational modifications don't happen in the thymus. So you've got to tolerize them extrathymically. That's called peripheral tolerance. The liver plays a particular role in that. So the liver circulation is very closely connected to the gut circulation. So there's exposure to antigens from food, from the commensals in the gut. Moreover, the liver is an enormous filter. It's a many functional organ, but it's an enormous filter in clearing apoptotic debris. And so, we're obviously dying all the time by apoptosis. And why don't we develop autoimmunity to all that debris? We believe, and I think we've got data, and probably others too, saying that the liver is very active in maintaining peripheral tolerance to those apoptotic debris. And moreover, there's some really old hints. So when people transplant surgeons first started doing transplantation, it turns out the liver is easier to transplant and not get rejection than most other organs, which was sort of like a hint, that there must be something tolerogenic going on in the liver. So the liver is, how does it clear? How does it clear apoptotic debris? It scavenges them maybe in a number of ways, but the one that we're particularly interested in is by changes in the glycome, of the surface glycome of apoptotic debris. So neuraminases get activated by apoptosis, and that leads to cleavage of the normal terminal sialic acid that you'll remember on the end of cell surface glycoproteins to expose N-acetylgalactosamine. Or with other proteins to expose N-acetylgalactosamine. There are then C-type lectins present on many cell populations in the liver that can collect via receptor-mediated endocytosis and lead to antigen processing through those C-type lectins of that glycome. And there are many interesting cell types. There's a specialized macrophage population. Those are antigen-presenting cells. There's a dendritic cell population. One that was particularly interesting to us was an endothelial population called liver sinusoidal endothelial cells. And moreover, hepatocytes can do this themselves too. And Martina Damo and Scott Wilson published in the lab that hepatocytes can even collect antigen and present it on exogenous antigen and present it on MHC1 to induce tolerance. So what we want to do is say, gosh, and Scott and I sitting around in a bar called the Satellite, drinking. They served specialized in Belgian beer, so we were drinking Belgian beer, and we were just like, how can we do this? Well, we can just synthesize that, can't we? I can't, but Scott can. We can mimic that. We can mimic the glycome of apoptotic debris pretty easily. So here we used a polymerization scheme with monomers that were glycated and monomers that were hydrophilic in spacers. And in terms of glycation, you could think of using something complex like a branch saccharide. We didn't think that was necessary to start with, and we just used N-acetylgalactosamine and N-acetylglucosamine, and later I'll show you we've gone on to mannose. So you can make a glycopolymer that looks like the surface of an apoptotic debris. And then we connect it. We connect it to the antigen. So the antigen is a protein or a peptide. And what you want to do in connection is in the endosome, you want that antigen to be released. You don't want to destroy the antigen by conjugation. So here we were just conjugating to lysine side chain, to the primary amine on the lysine side chain. But we did it, Scott did it, with a self-immutable linker, such that a disulfide that you'll see there in the linker gets reduced in the endosome. The free thiolate comes back and attacks to liberate the native amine on the side chain of lysine. And that happens, we showed, like in a period of like 10 minutes after endocytosis. So anyway, I'm going to skip the immunology. There was like painstaking 4 years of immunology to get to the animal experiments. So Scott published this first in a Nature BME paper in 2019, where we showed really good results in prophylaxis, and then some hints related to therapy. But then a bit later, a few years later, Andrew Tremaine and Rachel Wallace, together still with Scott Wilson, looked at therapy. There are autoimmune models. There are autoimmune models of psoriasis, of multiple sclerosis, sort of, call it neuroinflammation, of type 1 diabetes, sort of. And none of these models are perfect, but it's kind of what you got, and so you don't have much choice about otherwise. So this is a murine model of, call it multiple sclerosis, and more precisely of neuroinflammation, where the animal is immunized against a myelin protein. Those are the proteins that are the targets in multiple sclerosis, this autoimmune, the demyelinating autoimmune disease in the brain and the spinal cord. And so in the saline group, in the blue, you'll see as we induce the model, an autoimmune response that leads to a clinical score. The clinical score is a functional score of paralysis, basically, and it's highly nonlinear. So an animal with a score of 3 is like dragging itself about by its forelimbs, with its hind limbs basically non-functional. An animal with a score of 1 doesn't flick its tail quite like normal. So that's what I mean by quite nonlinear. So in this model, it mimics some features of the major form of multiple sclerosis called relapsing remitting MS. And so you'll see the score goes up. So pointer doesn't seem to work. The score goes up, it comes down, and then over a period of time, it relapses. That's what happens in patients. FDY720 is a clinical drug called fingolimod or galenia. It's an S1P receptor modulator. There are like a bunch of S1P receptor modulators. This fingolimod was the first one to be introduced, and it's still kind of standard of care. So there you'll see in the gray line at the bottom when we were dosing, sort of as a benchmark. And it acts in a totally different way. It acts by trapping lymphocytes in the lymph nodes in the spleen and not allowing them to go into their target tissues. So it's a broad immune suppressant by blocking cell migration. And it's quite effective. It's a good drug, but if you're on it and get vaccinated, you don't really have much of a response to vaccine. So it's a substantial immune suppressant, and we were thinking about, and not antigen specific at all. And you can see that when we stop treating in the gray line at like day 37 there, the disease comes back with a vengeance. So you didn't actually remove the disease, you merely suppressed the disease while you were on therapy. Okay, so what? So others have tried therapy with the antigen itself, sort of like the analog of the sub-Q thing I was talking about with environmental allergens or food allergens, just antigen therapy. And the antigen here is called PLP. It's also active, involved in the human disease. And you'll see in the gray arrows with PLP up top, we gave 3 doses, so that you could call it a vaccine, but it's not like got all the polymer bit and liver targeting stuff on it. The animals relapse then, so the red line comes back up to the blue line. So the animals do relapse. It's not effective at inducing a response with memory. And so what we were really thrilled is this P-gal, we call it, the N-acetylgalactosaminoylated polymer, conjugated to the PLP peptide via a self-inhibitor linker, led to really stable remission of disease. So we were really very happy about that. The tails didn't flick quite like normal, but it's a lot better than a score of 2, which is like serious waddling, a very clear motor dysfunction. So we're really happy to see that in therapy mode, and Andrew and Rachel published this in Nature BME in 2023. Not to brag for my lab, but to brag for them. I never look at those metric buttons, but for some reason I did. I couldn't sleep one night. But in the second week of publication, so it had like 14,000 downloads in 2 weeks. And Nature BME says it was like in the 99.9th percentile of attention. I couldn't believe it. Whatever, you get lucky sometimes. So we're really happy. How do you drive it forward? And I'd like to talk about some clinical results that come out of a company that I started to drive this technology forward. And the first human concept, and I would point out the asterisk, that I'm not implying that the exact molecule that I showed you on the slide is the molecule that was tested in the clinical trial. It's the same concepts, and there may be differences in the details that are not disclosed. We're interested in celiac disease. Celiac disease is an autoimmune disease associated with eating gluten. There's gluten insensitivity, which I believe exists. That's not celiac disease. But then there's bona fide celiac disease, which is you can very clearly immunologically diagnose. The antigens are really well studied in human celiac disease. It's limited to 2 HLA alleles with like 80% or 90% in one HLA allele. So it's a super very clean, as it were, autoimmune disease. So it leads to villus atrophy, obviously GI symptoms, and there are clear readouts that you can make. So this was the clinical study that published this year. Note that I'm not an author on this. These are the clinical teams and the drug development teams of this concept in the context of celiac disease. So published in Lancet Gastrohepato, and I'm a shareholder. So I could be, I can't be lying because this is peer reviewed by other authors. So anyway, why celiac disease? I mentioned it's inbred, as it were, limited to certain HLAs. So moreover, you can challenge the patient. So you can take a patient that you know immunologically has celiac disease, very defined. You can dose them, and then you can challenge them. You would love to give them a piece of Giordano's pizza. Actually, that stuff is awful, but you'd love to give them a piece of good pizza. But in fact, you feed them a really nasty gluten shake, which has a more precise dose. And you can measure then all sorts of things from them, from their blood. So in here, we were looking at, or the team, the clinical team, was looking at immunological cytokines, inflammatory cytokines in the blood that are indicative of immune response upon ingestion of gluten. And so the placebo in the blue bars, the left-hand side is interleukin-2, the right-hand side is interferon gamma, 2 pro-inflammatory cytokines. And you don't actually need much of a statistical analysis to see that the concept seems to hold together. And there are other data as well, and I've been shocked to see how much the mouse correlated with the human when we looked at the details of the immunology. It gives me a little bit of hope in sometimes otherwise hopeless days. So this has gone on into a phase 2 study now, and it's also that same concept, again, not necessarily molecule, is in a phase 1 in multiple sclerosis, which is a much longer trial, a much more complicated trial. I'd like to mention something else we're working on in the context of inverse vaccines, and that's allergy. So we a paper in food allergies in press, in Cell Reports Medicine, but work that's not yet published in the context of allergic asthma is being led by Immunoengineering in the lab in collaboration with Melody Swartz. And this was, I thought, a particular challenge in that the animals or the humans are already sensitized, and moreover, they're hot and heavy sensitized with IgEs, which are unmasked cells, and when they encounter the antigen, they cause anaphylaxis. So if I'm thinking about all those injections are IV, I didn't say that. If you think about an IV injection of something where there are IgEs, that's probably a bad idea. But going back to the previous talk, thinking about brushes and steric stabilization, I didn't say it, but on that antigen are like 10 of those polymer chains. So there's a steric shield around that. So antibodies like IgEs can't bind through that steric shield. But I can still get antigen processing after endocytosis because all that steric stuff pops off and liberates the antigen. So in this case, we're interested in asking, can we modulate the immune responses in an allergic animal? And this was in the context of allergic asthma. About 2/3 of asthma is caused by allergy. allergy to environmental antigens, allergy to house dust mite, about 60% of those are house dust mite. And so I'm going to skip all the immunology. This paper is in review now. It'll be trashed soon, I'm sure. So the animals are sensitized. They're allergic. So Immuno gives them 2 doses there in the purple lines at weeks 3 and weeks 4, and then challenges them intratracheally. So you expose them to the antigen. And so here on the left, in the left image, this is looking at inflammation. These are inflammatory. So sort of dark blue means inflammatory stuff. But looking outside in the parenchyma of the lung, in the upper left, you see not much inflammation. So what do we do if we don't treat it? If it's just allergic and we don't treat it? In the upper right, you see a lot more inflammation. There's a lot more blue. So that's bad. But in the lower right, that's in the case of the tolerized one. You don't really see much inflammation there. And when we quantify it, it's quantifiably different as well and statistically different. Quite encouraging. So if we look at mucus was also mentioned in the previous talk as being a barrier for delivery in the lung, but mucus is hypersecreted in these allergic asthmatic situations in the human and as well as in the models. So the mucus is stained here in sort of dark red. So you see not much mucus in the upper left in the healthy airway. In the right, on the left side of the right, you see gobs and gobs of mucus. That's really bad. But in the lower right, you see not much. Again, statistically supportable. So, and this was in the context of the sensitized animal again. So we're undoing sensitization. So you can even, I mentioned how dangerous IgEs were, but even looking at IgE responses. So the purple is the tolerized animal. And we're looking for purple being lower than the other scores. So upon challenge, those multiple blue arrows coming down to the top, you see that the IgE levels are substantially lowered. And even if we re-challenge, they're still substantially lowered. And even if they're re-challenged again, they're still substantially lowered. So, 110 a day experiment, I don't have that much patience. But, so yeah, you say that this has durability. And I want a tolerance response that's durable, meaning it has memory. And Emiliano showed, and that I won't show you, that this is driven by regulatory T cells. So I'd like to go to one brief example. I've got about 8 minutes left. Thinking in cancer, we've worked very closely with Melody Swartz's lab in cancer vaccination in a variety of contexts. And so in cancer, we'd like to vaccinate against those autoantigens that are mutated proteins. And so there are adjuvants that you usually use in vaccines, like toll-like receptor agonists. They're one class of antigens, and they're useful in that they can induce the induction from dendritic cells, antigen-presenting cells, of pro-inflammatory cytokines that trigger T cell activation. In this case, we're particularly interested in CD8 T cell activation because they can kill. They don't protect, they kill. And as well, some of those TLRs can even induce directly apoptosis to get sort of a double bang for the buck. So how could you inject something in the blood to bind to a tumor cell with these agents on it? People have tried that. Antibody drug conjugates are a big thing. People tried antibody conjugates to these toll-like receptor agonists. The problem is you just made a vaccine against the antibody. So you get an immune response that makes the antibody not useful after a couple of injections. And so Anna Slezak in the lab, and so we started thinking about, would there be maybe non-protein-based binders that we could use? And so it turns out, as you would know, that extracellular world, so almost all pairs of cysteines are disulfide bonded. But tumor cells do upregulate an enzyme called thioredoxin that leads to particularly low fidelity disulfide bonding. So the tumor cell is really decorated with a strong nucleophile right there, sitting in your face, with free thiols. And so Anna first published a paper in ACS Central Science last year, so where she made, and I'll show you the chemistry in a minute, but polymers that are cysteine reactive that contain one of those toll-like receptor agonists, a TLR7 agonist. You see it there. We picked it just because we can synthesize it, so it's something that's synthetically tractable. And she got quite a nice response with intratumoral injection. But we wanted to go after liquid tumors and ask whether we could vaccinate against liquid tumors. And so liquid tumors may be where the high frequency of free cysteine on the tumor cell compared to the normal cell would give us enough selectivity that we wouldn't kill the animal with off-target effects, but rather we would vaccinate using all the antigens. And I'd like to add a parenthesis. Cancer vaccines, there are a number of approaches. One is to sequence, determine mutations, predict which of those mutations are going to be immunogenic, either put them in an mRNA vector, Moderna has a trial that way, or synthesize them as peptides and make a personalized vaccine that way. That's usually against one antigen, but I mentioned that it can just downregulate that antigen and escape. So but this concept of Anna's is saying, like, if I decorate the whole tumor cell that's got maybe 100 mutations in it, every one of those hundreds is my vaccinal antigen. So it's hard to downregulate so many. So this is the polymer, simple pyridyl disulfide on one of the monomers, toll-like receptor agonist. And moreover, I'll go back to C-type lectins. So there's a mannose functionality here. Dendritic cells have a mannose receptor that's involved in collecting microbial debris to induce an immune response on it. So the polymer here is functionalized for dendritic cell targeting, for dendritic cell activation, and for antigen binding via the cell. So it's looking quite promising. The work is still ongoing, but this is an AML model. Quite interested in AML. My mother died that way. You can transfer AML cells in the mouse. This is the immunocompetent mouse I'm talking about. So all the animals die, and this timeline shown in the black. Cytarabine is a chemostatic agent. It's a standard of care for AML, and you see that it extends life somewhat. Interestingly, cytarabine upregulates the activity of thyroidoxin. So the cytarabine treated cell, AML cell, is like hyper then thiolated, with this strong nucleophile there. So it really binds the polymer. And you'll see this vaccinal response, this benefit that gave the red line when we combine the cytarabine with the mannose-related TLR7 containing pyridyl disulfide binding polymer. Anyway, that's a vaccine. And we have other approaches to vaccines. Cancer vaccines, we're quite interested in that area. But I'm quite excited about this one because it's so simple. It's just a polymer, and you just inject it in the blood. There you go. Anyway, it's kind of fun so far. So anyway, I'd like to close with daydreams. I try to spend time daydreaming about science, not about the weather or a beach, but daydreaming. And we as a team, I mentioned in a little video the team sport bit about invention. We spend time together daydreaming. We schedule time to do it. And so the inverse vaccine, I'm really super excited about that. Very interested in anti-drug immunity. We have a paper in press in Cell Reports Medicine, other stuff cooking in the lab. But there are many drugs to which the patient is immune. There are microbial enzymes that are used, for example, but moreover, like if you consider a kid with hemophilia A, they have no factor VIII, they don't express it, they didn't express it in the thymus. When you give them the factor VIII, many of them develop so-called inhibitors, which are antibodies, and then they're well and truly in trouble. And there are a whole host of diseases, congenital diseases like that. So we'd like to be able to do something to prevent that, and we're working on that quite hard. I mentioned already, thinking about simple platforms for nucleotide drugs or protein drugs that can be stored dry. And so there we're working with Stuart Rowan very closely at the University of Chicago on non-refrigerated polymer micelle systems that can encapsulate a really high efficiency, both protein, which is interesting, but nucleotides as well. So I think that's a major challenge. As I said, the mRNA vaccines impacted us a whole lot, but people in sub-Saharan Africa, it was the Oxford vaccine the viral vaccine that impacted them. So another context not related to polymers, where our immuno-oncology efforts are really more focused on, or highly focused on, cytokine targeting the tumor microenvironment, chemokine targeting the tumor microenvironment. We're interested in anti-inflammatory cytokines. I mentioned this inverse vaccine. I'm out of time now. My inverse vaccine targeting autoimmunity in an antigen-specific way. I don't know. I just want multiple shots on goal. I'd like to have an antigen non-specific approach too. And so we've identified the lymph nodes as a particularly interesting target, published a bit, and have work coming on out right still on anti-inflammatory cytokine targeting to the lymph nodes. And then as well, very interested in chronic inflammation. So not just autoimmunity, but targeting immune cells in chronic inflammation. So with that, the team, the sport best played as a team. I'm deeply grateful to my team. I mentioned that little video that I'm just amazed at how inventive and dedicated the people in the lab are. I mentioned other members of the broader team, meaning collaborators. The cancer work that's gone in the lab has been almost all with Melody Swartz, who's another chemical engineer like me, but more of a card-carrying tumor immunologist. The allergy work in food has been with Catherine Nagler, a mucosal immunologist. Some of the protein engineering work that I didn't mention with Juan Mendoza, really strong protein engineer. Stuart Rowan, well known to most of you, I think, polymer chemist. Matt Terrell, we've done a fair bit in nanomaterials with Matt Terrell, and I very much appreciate that together with Yun Fang and James LaBelle. With Jun Huang, we work closely in T-cell engineering, which I didn't talk about, and Sihang Wang on implantable materials and modulating inflammation. Lastly, that's science. I'd like to also mention, since my arrival in Chicago in 2014, really collaborated closely with Matt Terrell as dean. He was in the video. So with Matt Terrell as dean, as leader, building up nanotechnology at the UChicago campus, building up immunoengineering together with Melody Swartz at the UChicago campus. And really very much, there's a bunch of financial sponsors I won't mention, but very much mentioned the support of people with shared passions like David Kabiller and the IIN for driving this broad science forward. With that, I think I ended up like a minute and a half over. Thank you very much.